Hi, good morning, everyone. This is Xiaofang Wang. I'm the founder and the chief technology officer of ImStem. And this is a forward-looking statement. And this is a company overview. And we are a clinical stage biotech company, so focusing on developing stem cell treatments for rare disease with unmet medical needs. And our key technology is called TMSA technologies. And with this technology, we have the first-in-class human ES-MSC cell therapy product. And after many years of hard work, and we received the FDA green light to advance the product into phase 1 to 8 open-label clinical trial to treat multiple sclerosis. And the trial initiated in August last year, and we so far dosed 8 patients, and we expect to have the first data readout by the end of 2023. At the meantime, we also plan to file another IND targeting cerebral adrenoleukodystrophy, the so-called CALD, and in collaboration with University of Minnesota, and who is also behind the STARBEAM clinical trial of Bluebird. And we plan to file that IND applications by the end of this year. And here is our experienced executive team, and other than me, all of them have very good industry background and very good experience in develop rare disease treatments and neurological diseases treatments. And the TMSA technology is also a technology platform. And with this technology, we can generate high-potent MSCs from pluripotent stem cells like iPS, human ES. And this technology enable us to make the first in-class off-the-shelf allogeneic stem cell therapies, and with mass production capabilities and consistent qualities. And it also shows very safe and efficacy profiles in preclinical studies. And these therapeutic products also have the potential to treat many rare autoimmune diseases, as well as some inflammatory and degenerative disorders. And here is a key advantage of the TMSCs. TMSC is a unique kind of human ES-MSCs with high purity and qualities. And these TMSCs have a high potency in immune regulations compared to the bone marrow MSCs and also other adult MSCs. And these TMSCs can easily pass through the blood-brain barriers and can show some repair functions to repair the damage of the BBBs. And lastly, these TMSCs has a capacity for mass productions and without any concern of scalability issues that a lot of the adult tissue-derived MSCs has facing. So to elaborate a little bit more, and this shows that the TMSC was differentiated from human ES cells through a two-step differentiation method. And the first step is to differentiate the ES into a trophoblast-like stage. And through this stage, we can differentiate all the ES cells into trophoblast, almost 100% differentiations without any contaminations or remain undifferentiated pluripotent stem cells. And after that, we differentiate the trophoblast into the human ES-MSCs, and we can generate homogeneous cell populations without any off-target cells you don't want. And the TMSCs also shows much better lymphocyte suppressions in vitro, and we have some publications and show that our cells suppress the CT8, CD4 cells better after the CD3 stimulations. And we have shown our cells has better B-cell suppression activities compared to the bone marrow MSCs. And other than that, we also found that these cells have distinct cytokine expression profiles. First is express a high level of PD-L1, TGF-beta compared to bone marrow MSCs. Another important thing is these cells express very low level of L6, L8, IP10, and CCL2, a bunch of pro-inflammatory cytokines. And also, this cytokine expression will not upregulate upon the interferon gamma or TNF alpha stimulations, which very often you will see in an inflammatory milieu. And also, the TMSCs, which have shown that has very good penetrations through the blood-brain barrier. And as you can see here, the GFP-labeled human ES MSCs can penetrate and spread into the brain parenchymas pretty good, but the bone marrow MSCs will only accumulate in the blood vessels. And we have shown in vitro to confirm this, that the cells can perform penetration in transval assays to show that the BBB penetration is very good. And also we have shown that TMSCs can repair the BBB damage through the upregulation of those tight junction proteins like CON1 and CON5 and ZO1. And another thing that prevents the adult MSCs to become mainstream is because there are scalability issues. And with this new technology to differentiate MSCs from ES cells, so we have no problem with the scale mass production anymore. And we can differentiate the ES cell into TMSC in less than 6 to 15 days. And after another 30 days of expansions, we can generate a huge amount of TMSCs. Basically, it's with one donor of ES cell bank that we licensing, we can generate more than 50 billion doses of treatments to supply all the global needed. And here is the IND enabling studies we used for our IND clearance. And this is shown that once the mice was induced EAE, and this is called experimental animal influenza myelitis, and this is a typical model for multiple sclerosis. We have shown that the cells can reverse the disease course. Actually, when the mice are already induced paralyzed, and after injection of the cells, it can reverse the symptoms, and the mice can move around the cage with no problems. And this is the safety studies that show these cells after long-term engraftment into the mice, the immune-deficient mice. There's no tumor growth. So with this, we gained the FDA green light to going into the phase 1/2A clinical trials. And this clinical trials is for 30 patients with 3 cohorts. And the first is 10 patients with low dose. And the second, then we dose a high dose patient with another 10 patients, and then we will repeat the high dose after 6 months. So far, we have dosed 8 patients, and the overall shows well, safe and tolerated, and justify for continued enrollment. And which is also encouraging that we see some patients and have some anecdotal positive feedback, show some symptom improvements. And we are very excited about those results, and we will have the final data readout by the end of next year. And here is our summary of our pipelines, and our leading indication is for multiple sclerosis, and we are also focusing on other rare diseases like CLD and ARDS, and we plan to start a CLD trial next year. Here is a final summary of our investment highlights. And ImStem is focusing on rare diseases. Although we have leading indications, multiple sclerosis, we are seeking a partnership with larger pharmaceutical companies for those large indications. And we have our innovative first-in-class human ES MSCs. That is the first human ES MSCs gained the green light by FDA in US. And we have all the proprietary TMSC technologies can generate high-potent MSCs from pluripotent stem cells. And these cells have demonstrated its efficacy and safety in animal, and potentially efficacious and safe in human. And lastly, these cells can be manufactured at a large scale and cost-effectively consistent in mass productions. And thank you. That is my presentation. Thank you.